Okay, well, thank you. Thank you for the introduction. I actually changed the title of my talk a little bit just to, just to switch things up. Yeah, first I wanted to thank the organizers, Laura, Alexandra, and Vittoria. This is an incredible workshop, and I'm jealous that you're all in Vienna. I'm sorry that I couldn't be there in person. Logistics didn't work out, but I'm happy to have this opportunity to share some of what we've been doing with you all, and I hope we can continue to cross paths in the future. As was mentioned, I've been here at Princeton for the past 7 years, but I'm actually moving to Caltech this fall. So Christina was at Princeton and then moved, eastward. I'm at Princeton and I'm moving westward, and so yeah, we're balancing the center of mass. So today so Christina gave this incredible talk talking about single bacteria moving through motility. And so today I'll tell you about some of the work we've been doing, looking not at single bacteria, but collectives of bacteria. And we're looking at not how they swim, but actually how they grow. But hopefully what I'll convince you of in the next 25 minutes or so is that there are really interesting questions relating to transport of soft matter systems when we think about how microbial colonies grow at these collective scales, and how similarly we can use ideas from transport phenomena and fluid mechanics to actually learn something new about biology. And so even though, my group has studied single bacteria swimming in many different ways, today I wanted to share with you something completely different, because I think people in this audience can make really important contributions to this problem. And so why do we care about microbial growth? Well, for starters, and I think Christina introduced this beautifully as well, typically in the lab, people study microbial growth, in test tubes and bulk liquid cultures, or on flat surfaces, on Petri dishes. But in reality, in nature, right, natural microbial colonies grow in complex spaces. Christina mentioned some examples of this. They grow in the soil, in the ground beneath our feet. They grow in our bodies. They grow in bodies of water. They grow in industrial reactors. And in all of these cases, as Christina mentioned, we have chemical heterogeneities. Typically in 3D, you have mechanical interactions, you have fluid flows, you have interactions with various macromolecules. And these are all complexities that we've been systematically studying in my lab, both experimentally and using theory and simulation. And in addition to that, you typically don't encounter just a single cell type, but in nature, we typically have mixtures of different species and strains that coexist, right? In dental plaque, in our intestines, in natural communities, you typically have different cell types that have different chemical requirements and metabolic requirements, et cetera, right, that inhabit the same space. And so this also is an additional complexity that we need to think about when we would try to make sense of the physics underlying these kinds of colonies. And so the overarching question that we've been addressing is, how do these complexities shape microbial colonies? And why do we care about shape? We care about shape because it's known in biology that the shapes of colonies influence how they resist external stressors, how they grow overall, how they maintain genetic diversity. And so the punchline of what I'll tell you about today is that over large lengthened timescales, and I will clarify exactly what I mean by large, growing microbial colonies, even though typically we don't think about growth as kind of a transport process, we can think about these growing microbial colonies as self-generating, chemically responsive fluids. And I think that conceptualization gives us really powerful ways to learn something new about biology, but it also motivates new transport questions. And so just in the interest of time, I'll just focus on these two complexities, chemical heterogeneities in 3D and having multiple cell types, but we've studied these other complexities in various ways. So first, before I tell you about the science, I want to tell you about all the people who made this work possible. I unfortunately cannot see the audience, but I hope you're all pulling out your phones and scanning these QR codes. These link to the two papers that I will tell you about today. So Laura, I hope everyone's pulling out their phones right now. So the people who made this work possible, all the experiments that I'll tell you about today, were started and led by a former postdoc in my lab, Tapamoy Bhattacharjee, who's now a faculty member at the NCBS in India. The experiments were continued and pushed along by Konane Bey, who's now a faculty member at CU Boulder. Anna Hancock is a graduate student in the group who also made important contributions to those experiments, along with Haoni Liu, who was an undergraduate in the lab. She's now a graduate student at MIT. Alejandro Martinez Calvo and Carolina Trinado Yuste have led the theory and simulations that I'll tell you about. And Alejandro, Carolina, and Mira have also made further contributions to the experiments, and I'll tell you about that in a bit. And then finally, we do this work in collaboration with our friend and colleague Ned Wingreen, who's also here at Princeton. And we also have an active collaboration with Han Seok Lee and Jeff Gore at MIT. And I'll mention some of that work. And so the work that I'll tell you about today, this represents, a small subset of our more recent interests. An early version of this came out in this PNAS paper recently, and then we have a new bioRxiv preprint building on this work. And so these, please check these out if you're interested. Please take a look at our lab website. I'd love to hear from you and hear your thoughts. So first of all, I need to tell you about how we did the experiments that we did. And so all the experiments that we've done in my lab on this project rely on this very simple experimental platform. What we do to study how microbial colonies grow in 3D versus on a 2D flat surface is we use what are called granular hydrogels and granular hydrogel matrices. What we do, so these granular hydrogels are grains. They're internally cross-linked networks of hydrophilic polymers that really like water and are biocompatible. And so when you take one of these dry granules and disperse it in whatever liquid cell culture medium that you want to use with the right nutrients and salts for the cells, that granule swells up, it absorbs all that liquid, all those nutrients, all those salts, right, until it can't swell anymore because of its internal elasticity. And so if you add enough of these dry granules to your liquid culture, then they swell up and they form a jammed packing, right? Like a ball pit for the cells. If you add more of them, you get a tighter jammed packing where the pores between the grains are smaller. If you add less of them, you get a looser jammed packing where the pores between the grains are bigger. And so this way we can tune very precisely the structure of these matrices, the elastic properties of these matrices. And what's important about this is that these are completely transparent because the hydrogel is so highly swollen, right? So we can actually spy on what the cells are doing inside these 3D matrices. These matrices have very low yield stresses. So even though they hold the cells in 3D, they don't impose strong mechanical confinement to the cells. And because they're so porous internally, right, oxygen and nutrients, which are much smaller than the internal mesh size of these hydrogel grains, can diffuse through completely unimpeded. So we are physically holding the cells in 3D, but we're still enabling them to access oxygen and nutrients in 3D. Right? So this is our experimental platform. And best of all, one of the things that we realized we could do, inspired by work that other people have done, is we figured out that we can 3D print microbial colonies inside these granular matrices. So we can literally put whatever cells you want, wherever you want in 3D, right? We're 3D printing microbiomes. And the idea behind this is very simple. I told you that these are jammed packings, right? They have a yield stress. And so just like a typical jammed packing, what we do is we take an injection micronozzle. So we built a 3D printer in my lab. We have an injection micronozzle that goes into a hydrogel matrix. It extrudes cells from the tip along any 3D path that you trace out, that you program. As it moves, it spits out those cells, it pushes those hydrogel grains out of the way, and then they repack around the cells and they hold them in place, right? So this is truly 3D printing inside these hydrogel matrices. People have used similar approaches to 3D print tissues and organoids. We've now adapted and hacked this approach to 3D print microbial colonies, right? And so we've had a lot of fun engineering different kinds of microbial colonies using this approach, using these granular packings. We've looked at how single cells move and do interesting things, right? Christina talked about some of that, right? And some of the references are shown here. We've also studied how collectives of these cells move through using chemotaxis through these hydrogel matrices. We've also kind of studied that extensively. But what I'll tell you about today is for the case where the pores of these matrices are so small, they're smaller than the body size of the cells, so that even if the cells are motile, right, they can't get anywhere. They're stuck, and now the only thing they can do is consume nutrients from their surroundings, grow, push out on the pores. Their turgor pressure is large enough to deform the pores, and push their progeny to the next pore, and so on and so forth, right? So in these hydrogel matrices, we can tune the relative importance of motility versus growth in contributing to the overall spreading of a colony just by tuning the pore size. And so now we're in the growth-dominated limit where we have these individual cells that push out through growth, they push their progeny to the next pore, and so on and so forth. So we're just looking at growth, right? And so what we did is we said, well, we want to understand how bacterial colonies grow in 3D. So let's 3D print the simplest possible thing we can think of. What you're seeing here is a dense packing of E. coli. They're all fluorescent, right? That's been 3D printed in a cylinder that's embedded within one of these hydrogel matrices. So what you're seeing here is just a small field of view. The cylinder is much longer than what you see here. This is pretty zoomed out. Each pixel is a single cell. So the entire width of the screen is thousands of cell body lengths, right? And so we have this cylinder that's embedded within this hydrogel matrix. We're taking a slice through its midplane, right? So we do 3D imaging, and you're looking at a slice through its midplane. So this is the initial condition, right? We have this cylinder, dense packed E. coli, sitting in this hydrogel matrix. And the question, and the matrix is so tight that the cells cannot swim, they can only push out and grow. And so the question is, how will this initial inoculum grow in 3D? And so here's what we saw. So as these things grow over 100 hours, they form broccoli, right? They don't just spread uniformly outward, but they grow into these beautiful multi-scale fractal-like patterns, right, in 3D that look like broccoli. We can characterize the structure of these extensively, right? We have resolution down to the subcellular scale, and we've done that, and they are indeed fractal-like at small length scales. But to use the analogy of broccoli, right, there's a characteristic floret size around 200 microns that bounds these fluctuations. And we kept seeing this over and over and over again when we did the experiments, over and over again, right? We tested many different hydrogel matrices of different pore sizes and stiffnesses and nutrient types and initial conditions. We always saw the same thing. Most importantly, we tested many different types of cells. We tested E. coli. We also tested Vibrio cholera, many mutants of Vibrio cholera. This is a gut pathogen and a biofilm former, same thing. We tested Pseudomonas aeruginosa, which is another biofilm former, and a lung pathogen and a soil bacterium, same thing. I really like kombucha. I don't know if any of you like kombucha. Sometimes when you drink kombucha, there's a sludge at the bottom. It's called a SCOBY. That's a biofilm. I convinced Konani to test one of the strains in kombucha just because I like it so much. We saw the same thing. This broccoli seems to be a generic feature of microbial colonies growing in 3D. And so the question is why, right? Why are they forming these broccolis? And so we can do a back of the envelope calculation, and that actually gives us a clue, right? It turns out this broccoli growth, as I call it, is reflecting a fundamental difference between growing in 3D and growing on a 2D surface. If you grow on a 2D surface, you can always access nutrients from the third dimension, right? But if you grow in 3D as a dense colony, inevitably there will come a colony size at which the cells on the outside are consuming all the nutrients and preventing nutrients from getting to the inside. And so the inner part of the colony becomes dormant. It becomes starved, right? And we can do a very simple estimate for how far nutrients penetrate before they're completely consumed by the cells, just by the competition between diffusion with a given diffusion coefficient, D, and consumption by the cells at a given maximal rate, K sub S per cell, multiplied by the density of cells, rho. C half here is the Michaelis-Menten a characteristic concentration. And when we do the simple estimate, what we find is that nutrients only penetrate 10 microns deep into these dense colonies, right? That's just a few cell layers deep. And in fact, we can see that with imaging. Metabolic activity is only localized to a thin surface layer. And then everything beneath that is just dormant. It's starved, right? And that turns out to be the reason why these broccolis form, okay? Why is that? So again, we can do this experiment on a 2D surface. And on a 2D surface, it expands smoothly outward. But then when it goes into the 3D matrix, it becomes rough and broccoli-like as well. So what's going on? And so how is this nutrient depletion inside the core giving rise to this broccoli growth? Well, we said, let's construct a model of this, capturing what we think are the essential features here. And so this is where the transport processes and fluid mechanics comes in. We usually don't think about growing colonies, right, as fluids, but we were inspired by an overdamped hydrodynamic treatment that people have used over the years, where over large length and timescales, we're looking at timescales much longer than the doubling time of the cells. We're looking at length scales much larger than the single cell scale. These are close packed colonies, so let's treat it as an incompressible fluid, right? What does that mean? What that means is we simply say that the colony is expanding outward at a given speed, right? Given by Darcy's law, essentially, right? So there's a growth pressure driving that expansion, right? And this fluid is generating itself. There's a growth rate G, right? That generates how much the new fluid, right? And so what's important, what we did that's different from previous models is we add in coupling to nutrient availability. So nutrient is given by C here, right? This function f of C reflects Michaelis-Menten kinetics of consuming nutrients. And so what we did is we coupled growth, right, to nutrient availability through this Michaelis-Menten function f, right? And so what's the nutrient doing? Well, it diffuses throughout the interior of the colony, and it's consumed by the cells, again, at a maximal rate K sub S per cell, multiplied by the density of cells rho, right? What's a nutrient doing on the outside? It's just diffusing. That's it. That's the only modification we made to this model. We've neglected many other complexities, and I hope that you all get interested in this and start to think about how these additional complexities change what I'm telling you about today, because they'll certainly be important, right? We've neglected any kind of mechanical interactions. We've neglected any kind of pneumatic ordering, any feedback from stresses, cell-secreted substances, nonlinearities. These are all interesting things to explore, but we said, let's just do the simplest possible thing. When we solve this model, we indeed find that nutrients only penetrate a small length that's much smaller than the overall size of the colony at long times. And so we asked, is it stable to perturbations? So we did a linear stability analysis. And what we found is that long wavelength perturbations, so small wave number, are unstable. And the intuition behind that is if you have a little perturbation, right, then it can access nutrients better and it grows faster. But small wavelength perturbations, large wave number, are stable. And the reason is because transverse growth can fill in these values. So we obtain this dispersion relation. And what's interesting is you have this most unstable mode shown by the red point. And that wavelength corresponds to 19 times this nutrient penetration length, right? That's it. And when we put in the numbers, that captures exactly the length scale of these florets that we see in these broccoli experiments. Moreover, when we do a numerical simulation of this model with noise, we get those fractal-like features as well, right? And so we have this competition between enhanced access to nutrients causing faster growth, but also transverse growth filling in the valleys and stabilizing these perturbations. And so we can do this in 3D as well. We get similar results. I won't go into the details of that. But what's nice here is we don't just see these broccoli things in the lab, but also in natural communities, in the ocean, in bioreactors. And this active fluid model gives us a way to understand the physics of why these things form. And everything I told you about was for single cells of a single cell type. What if you have mixtures? In more recent work, we started looking at mixtures of 2 different cell types. Initially, they're randomly mixed, but then something magical happens. They self-sort. They spatially sort themselves into the different florets. I challenge you to find a floret with multiple colors in it, right? You can't, because there's a founder effect where different cell types nucleate florets. And so this is something we're continuing to explore. But even beyond 3D, if we just go back to 2D, right, and look at the interactions between different cell types, and this was inspired by a collaboration with Jeff Kor's group, what we find is that when you have 2 different cell types growing on a 2D surface, in some cases, the interface between them can be smooth, right? In particular, it appears when the faster growers are on the inside and the slower growers are on the outside. But then if you flip them, we did this experiment, the interface became rough, right? If now the faster growers are on the outside, the interface becomes rough. And so the question is, how do we understand the structure of this interface? Well, we compiled even more literature data, performed even more experiments, and this seems to be a robust phenomenon, where in some cases, the interface between cell types is smooth. In other cases, the interface is rough. And so we said, can we use our similar active fluid model? But now, of course, you have 2 different cell types. So same thing. We're treating this core shell community, right, as a closed packed incompressible fluid. We're modeling it the exact same way, but now you have a difference between the 2 cell types. They have different growth rates or proliferation rates. So we can look at the difference between the growth rates, and they have different friction coefficients with the surface, underlying surface, which reflects, for example, differences in adhesion. And what we found when we used our exact same model that I just told you about, did a perturbation analysis of the interface, right? What we found is that when you have the faster growing on the inside, a grower on the inside, perturbations decay. This is stable. But if you have the faster growing on the outside, then these perturbations exponentially grow. This is unstable. You might think that this looks kind of like the Safman-Taylor problem, right? Because we're looking at these different kind of active fluids. This is not your typical Safman-Taylor instability. The dispersion relations, the growth rates are completely different from Safman-Taylor. And in fact, what you're seeing on the right here is that this interface is stable, even though the friction coefficients are matched. In Safman-Taylor, instabilities arise at the interface because of differences in friction between the 2 different fluids, right? So this is a different kind of instability. So how do we understand this? Well, why do these things become unstable? What we found through numerical simulations is that if you look at this outer interface in purple, right, at long times it becomes stable, but this inner interface between the 2 different cell types, it actually gets extruded. So you have the slower growers on the left, the faster growers on the right. If you have a little bump, the faster growers actually grow transverse and pull this finger out. And that actually causes this to destabilize, right? So this instability is arising because of this weird coupling between growth between the different cell types. And so using this model, what Alejandro and Carolina did is they constructed a morphological state diagram. On the x-axis is the ratio between the growth rates, between the proliferation rates, between the inner cell type and the outer cell type, or the outer and the inner. And the y-axis shows you the ratio between the friction coefficients between the outer and inner cell type. And what we expect from our model is that, and you can calculate this fully, is that in the bottom left, we get stability. We actually get M-dependent stability as we go upward, right? So that means different modes are stable or unstable until you get full instability in this top right, right? So now we can actually make predictions about when a microbial colony, the interface between 2 different cell types, will be stable and smooth or unstable and flat. And best of all, when we went back into the literature, when we went back to our experiments, and placed the different experiments on this state diagram, they were recapitulated. They fell exactly where we predicted. Stability was in the stability regime, instability was in the instability regime. And so even though I'm sure there are other complexities that we will need to take into account in many cases, at least for the experiments we have here, this simple model seems to capture the essential physics of what's going on. And so with that, I just want to conclude by pointing out that I told you about this incredibly simple active fluid model, right, that captures something about biology. But of course, building on this, it'll be useful and incorporate interactions, chemical interactions between the different cell types. They can secrete toxins, they can secrete metabolites. We haven't talked about mechanics at all or any kind of feedbacks on growth. These are all complexities that I hope you all get interested in and think about building on our very simple work to try to capture something about more complex microbial systems. And so we're excited because we think this provides an intellectual framework for understanding how microbial colonies get in shape. And again, the punchline here is that even though we typically don't think about fluids and transport processes when we think about slow microbial growth over large length and time scales, these ideas from fluid mechanics, right, are useful. And we can think about growing colonies as self-generating, chemically responsive fluids. And so with that, please check out these two papers. My group does a lot of other work studying active matter, bacteria, soft materials in other contexts. Here's our lab website. Here are the real heroes who make all this work happen. And I'd love to hear your questions. Thank you all for listening.